Alrighty, so I think we are live now. So welcome everyone to the Pakistan International Neuroscience Society Meet the Faculty Lecture Series. I will be your moderator for the session today. And then before we get started, I just wanted to go over some housekeeping rules. If you have any question during the presentation, feel free to type them in the chat box and I'll be more than happy to read it out loud towards the end of the presentation. Or if you just want to simply raise your hand, since we are a small group, we could do that, or unmute yourself, or turn on your camera, we could do that as well. And then I think without any further ado, it is my absolute pleasure to introduce Dr. Naweed Syed, who is a professor and chief scientist at the University of Calgary in Canada. So Dr. Syed completed his master's degree from the University of Karachi in 1984. And then he moved to the United Kingdom to do a PhD in neurophysiology. It is such a great and a pleasant coincidence that we both share the same enthusiasm and passion for neurophysiology. And some of you might know that I'm a current PhD candidate in neuroengineering and neurophys. So after completing his PhD in neurophysiology at the University of Leeds, Dr. Syed then moved to University of Calgary in Canada for his postdoctoral fellowship. And being there for 3 years, then he was subsequently appointed as an assistant professor in the faculty or Department of Medicine. And then he has been a professor over here since then. Dr. Syed has many national and international awards to his name. Personally, the most significant one to me is Tamgai Imtiaz from the government of Pakistan for showing his excellence in research and innovation. And talking about your scientific career, Dr. Syed has published over 150 peer-reviewed articles in the top-notch journals, so Nature, Neuroscience, Journal of Neuroscience, and Science as well. And then I must say that it takes a lot of patience and commitment to train the future generation of scientists. And Dr. Syed has mentored over 60 plus PhD candidates and also many postdoctoral and master's students as well. Well, I can go on and on about the great scientific career for Dr. Syed, but I just want to point out one thing, that I'm so privileged to find a role model in Dr. Syed who looks like me. Someone who we would not be able to find 20 or 30 years ago. And we are so much privileged that he is here with us tonight. And thank you so much, Dr. Syed, again, for accepting this invitation. And then the floor is yours now.
Thank you, Adil, so much for your kind introduction. And it really is a privilege. I think I see, young people like yourself who are enthusiastic, excited about neuroscience and field. And my heart actually, just starts thumping that the future is really bright. I think as an academic, nothing gives me greater joy than to see people who stood on my shoulders. They will see much further than I did at my time. So actually in you guys, we will live vicariously forever. So you actually make us immortal. So the credit goes to you guys, not us. We are just really chugging along and chasing our dreams, and I learned very earlier in my life to make a dream come true. The best way is to actually wake up. So it has been a wake-up call, and we do crazy things, and I'll be happy to share, some of our work. Because I gave a talk to Apna Group, and I don't know if many of you were there, so I will shift a little bit of gear. And since there are lots of younger trainees, I would really focus on work in progress first, and then I show you, what it is that we are heading in terms of neurons. So one's on the spinal cord injury. So if I could share my screen, there we go. Can you see my screen?
Yes, we can see your screen.
Okay, so first I will declare conflict of interest whenever I talk to my clinical counterparts. I have to do this. I actually have no financial interest whatsoever. All of my patents, even after I filed them, many of them licensing, we passed it on to Neuralink free. So everything is free. I don't believe in really hoarding knowledge, because I think that that is not conducive to any progress in the field, that you file a patent and protect and you sit on it. So no, I do have affiliation with board of directors of many companies, but I have no financial really benefits for all of that. So this is my city. I am from Calgary. It's beautiful here. We are having a heat wave, believe it or not. It's 32 degrees and everybody is panicking because it's a heat wave. So And warnings are coming everywhere that it could go up to 36 degrees, and that will be it. And somebody passed out by just hearing that it could be 40 degrees at some time. So, you know, doesn't really happen. So my research program, ever since I was an undergraduate, has really been focused on brain development. I've been fascinated, you know, there are about tens of billions of brain cells, more neurons in the brain than there are stars in the Milky Way and the galaxy. And how do they all connect together? How do these brain cells know where to go? And when they find their target, how do they connect? How do they recognize each other? And once they recognize each other, what allows them to develop synaptic connections and the connectivity between those brain cells? And what happens when these connections go haywire and they are disrupted either due to you know, neurotoxicity or environmental factors? And if that happens in the adult brain, how do you promote regeneration? So I have also been interested, in addition to neurodevelopment, in the area of neuroregeneration. And finally, brain chip interfacing technology. So what I'm going to do today is I will present some of the work from the lessons that we learned from brain development, you know, in the developing animals. And we will use both simple organisms, invertebrates, as well as vertebrate models, because they really, Mother Nature is very conservative. Mother Nature has used building blocks. So a neuron is a neuron is a neuron. A synapse is a synapse in a synapse, from a worm to fruit flies to snails to human. So those building blocks are just like brick. You can put it, you can make a, you know, a large high-rise building with it. You could build a mosque or a temple with it, or you could put it in your house. So a brick is a brick. So synapse is a synapse, neuron is a neuron. So the lessons that we learn from these simple organisms and model systems are really important, and because we need to go back to where Mother Nature, you know, used these technologies, different mechanisms to put this incredibly intricate brain together. So I will share with you today some work in progress, and that is in the regeneration, you know, arena, that how we are using brain chip interfacing technologies also to promote regeneration, and because in the peripheral nervous system, regeneration does occur, and we will use that as a model. And finally, I will talk about brain chip interfacing technologies as to how we can use those as that technology, not only to understand how brain functions, what are the connectivity patterns between the brain cells, what do they say to each other when two brain cells come together. And you know, you will see that when these brain cells chitter chatter, you begin to unravel the actual secret conversation that would be equivalent to a parent has got a chip or a hidden camera in the young kid when they go out for a party. So parents know everything. Or when you arrive at an airport, you will begin to see that you know, one person who's getting off in the airplane will come and say, how are you? So from the language, you actually begin to decipher as to where this individual is coming from and what their connectivity could be. So we will go in these three different areas, but first part of my talk, I will really primarily focus on developing biomedical engineering approaches to be able to see if we could promote nerve regeneration in the spinal cord or from the peripheral neurons. And then lastly, I will show you some of the evidence about how can we use brain-machine interfacing technologies or semiconductor silicon chip technologies to be able to A, record from large networks of brain cells, and B, if we could actually regain some of the lost brain function by using these technologies. And of course, you cannot really study any of this in the intact brain. It's extremely complex. You know, imagine tens of billions of brain cells. In a baby's brain, there are actually twice the number of brain cells when the baby will end up after birth and after a few months into birth. So half of those brain cells are killed because of the programmed cell death. So the actual brain cells in a developing brain are twice in number. And each and every neuron has hundreds and thousands of branches and connections. And it's incredible that not a single one ends up making a mistake. this really is absolutely magic. And that really keeps me, awake at times. So when you look at a simple, what we have learned over the years, we know that the neurons are the building blocks. These neurons are connected through specialized structures, call them synapses. Synapse is a means of communication between two brain cells. When they talk through, it's just like an embassy between Canada and United States being presynaptic neuron, Canada being postsynaptic neuron, and these synapses are the embassies through which they communicate primarily. And of course, they are supported by glia. Glia are no longer support cells in the brain that provide the livelihood and put food on the table, but they're also a very important player in terms of either myelinating the fibers to prevent the leakage of current. They're also important as a tripartite relationship between them and neuron is really critical. So these neurons, when they come together, they really establish neuronal circuits. And these neuronal circuits are highly dynamic and they undergo neuroplasticity. And this plasticity really allows us the ability to be able to, basically adapt. The brain is an adaptive machine. It's not like a computer that you think about, a hardwire. Imagine for all our life, the hardware is never upgraded. Like you do in computers, you buy a new, better computer, faster computer, in semiconductor technologies come in. But in case of these neurons, the brain being a computer, but it is highly plastic. It changes every time we learn something or any other external influence that comes into it. And those plasticity then really give rise to behavior, a different behavioral repertoire that really comprises the adaptability. So all of this is determined by, in part by the genetic makeup, but most of it really is epigenetics. And epigenetic plays a pivotal role in terms of really defining the final patterns of connectivity between these brain cells. So, in terms of monkey and a human brain, we're 98% of the connectivity in the brain cells, and even the genes are identical. But because in a monkey, the brain develops so fast that almost 75% of the brain is developed at birth, whereas in human, it's only 25%. So this epigenetics will play a very important role, the environmental factors that would allow slow and gradual really upgrading of the software as well as hardware. So here is a single brain cell sitting in a culture dish. Now this brain cell, imagine in your brain, and it has a remarkable task. Now imagine in a giraffe, a brain cell may be around the spinal cord. It has to go and innervate that little toe muscle. And this neuron is almost traveling across the Atlantic to be able to get to that place. And it has to navigate through this virgin territory. And it uses a number of cues. And those cues are really, first of all, the substrate on which it is growing must be conducive to growth. And then this brain cell will really get the cues from the environment, often diffused molecules, which could be trophic molecules that are released by target, or the adjacent tissue, like a road, whereby it is actually paved road. And so these neurons developmentally are guided by galvanotropic mechanisms, meaning that the applied electric field from the adjacent tissue allows them to go in a particular direction. And then the chemotropic mechanisms, whereby chemical molecules are often released from the target at a distance. And these growth cones, which are at the leading edge of each and every neuron, are really so smart that they can sense the environment and they grow in this direction. Now, in this particular case, you can see this is in seconds and minutes. You will see that the brain cell is growing all over the place because we gave it the trophic molecules that were present in the dish. So there was no selective gradient of trophic molecules coming from any given particular place. The other thing is that you could see that not only the trophic growth is everywhere, but also it doesn't have substrate adhesion molecules. We have coated this dish all along. So The entire dish is coated with laminin and fibronectin, or polylysine, the artificial charged substrate. So as a result, this brain grows everywhere. But this is not what happens in the intact brain. Brain cells don't grow in this fashion. Otherwise, they will end up really spending so much time. The brain will be mismatched, cross-innervation, and often you will turn out to be complete zombies. So a couple of things to remember, that neurons are guided by applied electric field. Even in an after spinal cord injury or nerve injury, you could detect actually electric current in the vicinity of the damage to nerve fibers. So we know that the tissue that is adjacent to the injured nerve generates applied electric field, which can be measured. So we know that this applied electric field is acting as a galvanotropic mechanism whereby the growth cone is recognizing that applied electric field. And if you do the experiments where you bring in a cathode and electrodes and anode, you will see the growth cone actually turn towards the positive molecules and turns away from negative molecules. So the charges. And then the trophic molecules that are released in a specific patterned way. Even in a spinal cord, you'll see there are gradients, chemical gradients that allow these brain cells to go from low concentration to high concentration. And this is how these growth cones navigate. So if you look at a single growth cones, and we have done a lot of interesting work whereby we can even cut a growth cone. And when you cut a growth cone, even a growth cone or a neurite that is cut from its cell body has the autonomous ability to be able to recognize both environmental cues. Absolutely amazing. You thought the brain was amazing, and neurons were amazing. Actually, these growth cones are absolutely amazing. Even if you cut it from the cell body, it still has the autonomous ability to be able to recognize its environmental cues, not only environmental cues, but also targets. So we know the molecular components of these growth cones. The red one is all actin molecules, and then green is microtubules. And you can see the filopodia, which is really the antenna, which senses the environment all over the place, are comprised of actin molecules. And when a particular filopodia moves forward, either it's pulled or it's pushed, the microtubule enter into that filopodia. And once it actually enters into filopodia, that becomes the leading edge of that growth cone, and it will turn. So the growth cone itself is really the brain within the brain cells that is endowed with incredible capabilities of navigating through virgin territory in the brain. And it does this autonomously because there is no way for a growth cone that is located far distance to be constantly in communication with the cell body if you think that the neuron's cell body is actually the major factor that makes all the decisions. So these growth cones will navigate through virgin territory. They go all over the place. So even if you use a culture model, these growth cones will grow in all directions. And I think the important thing to remember here is that when you think about nerve injury or a transaction, when the nerve is injured, we do know that as compared to the brain, the central nervous system, particularly the brain, where natural replacement of damaged brain tissue doesn't take place, regeneration doesn't occur, for the most vertebrate brains, and whereas the peripheral nervous system, peripheral nerves are capable of regeneration, they can regenerate. But what happens often is that this regeneration is often incomplete, and it is always almost accompanied by neuropathic pain. Because what happens is, now imagine that you are watching a hockey game or a Super Bowl, and if everybody were to leave at the same time, the traffic jam will occur. Nobody will go anywhere. So often happens is when a nerve is injured, all of these axons will turn on the speed to grow, but when they are all growing, they get mishmashed. They often get cross-innervated, and this cross-innervation leads to neuropathic pain and also incomplete regeneration. Now imagine that if you were to put traffic lights, and those traffic lights guide these neurons in a very systematic and an orderly fashion, you could actually get much better regeneration and also avoid neuropathic pain. So we know that the peripheral nerve injuries are often debilitating, and in most instances, they result in permanent loss of function. The peripheral nerves, as I said, they can regrow after injury, but the regeneration is often incomplete, or it's mismatched, or it is accompanied by neuropathic pain. So to achieve functional regeneration, and this is what I was talking about, work in progress, it is really important that we develop and design strategies that can facilitate functional regeneration. And then not only functional regeneration, but also to speed up the entire process. And we have published actually a paper in Neuroscience a couple of years ago, whereby we showed that this, we could 4 to 5 times, we could enhance nerve regeneration, and often using these metacarpal tunnel syndromes, the surgery, then the surgeons will, do their job, or if the nerve is resected somehow, we could actually enhance the growth by planting our portable devices, and I'll show you in a bit. But the other important thing is that when you're using a culture model whereby you're growing these neurons on flat substrates, and as we were talking earlier, that these neurons, when they grow on an artificial 2D substrate, which is plain polylysine dishes or laminin-coated dishes, the neurons get stuck to the dish or the substrate so tightly that if you introduce glia or the Schwann cells, these Schwann cells, for them to be able to myelinate a myelinating fiber, it has to get underneath. But what happens is that because the neurite is so tightly attached, these Schwann cells, they just hang around. They cannot get underneath to be able to wrap it and make it a myelinated fiber. And that is really a major problem for studying MS-related diseases or peripheral nerve injury, because these Schwann cells do not have access to axon, because axon is so tightly stuck. And if these Schwann cells, once they get underneath, it just like someone gets under your skin, all of a sudden, these axons get lifted up and they detach from the substrate, and then they pull back into. So what we wanted to do was to actually create a substrate which is 3D. So the 3D substrates, meaning that brain cells are actually grown in a substrate which is more like a jelly or a pudding. And we created Gelma, which is a interesting biocompatible substrate, and that allowed us to be able to grow these cells in 3-dimensional. So brain cells were no longer attached to substrate. So what happens now is that you introduce Schwann cells, and you could see that the process of myelination actually begins. And these axons begin to get myelinated. And it's really fascinating to watch. I don't have time to actually show you the videos whereby, a Schwann cells, they will interact with each other freely. But once a particular Schwann cell has made a contact with a nerve, and it starts to wrap around it, and you can see the wrapping around as well. As it starts to wrap around it, really the exciting thing is that it becomes repulsive to all other Schwann cells that will now contact it. So it seems like it acquires an identity, and it says, hey, this is my spot. You go somewhere else. I am engaged here, and this part of the axon is taken. And so we can actually watch them in the series and systematically myelinating, and we're using myelin-associated proteins and others. We can actually show that these axons get myelinated as we deem appropriate. So from a chemical engineering, where you can actually design these environmental substrate in a 3D, that allows you to be able to study myelination, which has really been a major problem in the field, because we know that there is really no cure for MS-related diseases. We also know we do not understand how axon get myelinated either with oligodendrocytes in the brain or Schwann cells at the peripheral nerve. But when you see these neurons, even in culture, the growth is really haphazard, it's random. And even if you grow them in 3D substrate, they still have the ability to roam around in the jungle. They just go around even in a 3D or a 2D substrate. So we needed to straighten them out. How could we really send a beacon to them and then guide them in a particular direction in an orderly fashion? So we developed, these chips, and these chips are interesting. In these grooves, we put down the electrodes, which are capable of, they are basically transistors coupled with capacitors. So some of these holes will have transistors, the other capacitors. And what we do is these electrodes can create an applied electric field whereby we allow these axons to act, for example, just like a tuning fork. The other thing we do is we take nanoparticles, we will dip these nanoparticles, we charge them with negative or positive charges. The core shell of the nanoparticle is charged, and then using the capacitive current, we will hold them to a particular capacitor. To give you an example, if the nanobead was charged positively, we will pass negative current through our capacitor, which is located here, to hold these nanoparticles or nanobeads. Now, these nanobeads are also coated with trophic molecules, for example, a BDNF, NGF, CNTF. Depending upon the cell type that we are using, we will coat them and we will hold them, we cage them. And then what happens is that when we want to release them, we change the polarity of the charge. So now the capacitor will, instead of having a negative charge, it will create a positive charge. So if the bead is negatively charged, it will be repelled immediately. It moves away from that and it diffuses along these grooves, and the growth cone or a neuron coming from the other direction will sense that cue and see the chemical gradient, and it will grow in that particular direction. So we put the traffic lights on these. So turn left, turn right, and we determine these neurons as to where they're going to go. They're not allowed to go in all over the substrate, but their growth is really targeted and directed and time controlled, as well as electrically controlled. So we can create networks of those same cells that you saw earlier. They were growing all over the place, but now we confine them and we restrict their growth to a particular direction and particular pattern that we define. So all these neurons that you see here are not growing all over the place, but they are, there are maybe a few that kind of try to get off the track, but for the most part, they will really grow only in one particular direction, the way they would normally grow in the intact brain. The growth is really not haphazard. And then what we can also do is we can mostly naturalize this process. We can take a particular neuron and we identify two of its targets. These targets always release trophic molecules because brain cells sense these chemical gradients and they grow in that direction. We introduce these two targets later, 12 hours later after plating the first cell. And when they release these chemical gradients of the trophic molecules, it's like a scent, it's like a smell, it's just like a syrup that is being sipping out of these. And then these, you can see that the growth cones can actually recognize and they grow in that particular direction. And what happens is that you can now create networks of these brain cells that you could define their patterns of growth and in a very highly orderly fashion. So the idea really is, moving on to how can we really now take it into a sort of a clinical setting. So I'm going to show you, we are working with Integra Life Science right now to come up with these neurotubes. And these neurotubes are meant to promote growth in a highly ordered and in a directional manner by using both chemotropic mechanism as well as using a applied electric field. So what happens is when you grow these cells, for example, in our culture dishes, what we do is that we could create, you know, that the growth cones are the one that we need to target because they are the navigator, they are the Columbus of the brain cell. And what we do is that we create a series of electrodes, which is in a tube configuration. These growth cones can sense these, applied electric field. When we stimulate this particular one single growth cone, will grow in the area that we are either uncaging these trophic molecules. So you can see that they're being picked up by the growth cone and it grows in that direction. We can stop and start. Now imagine that you do a tube whereby you have a series of these capacitors and transistors. You can, when a growth cone makes a contact with first series, it shuts off, it turns on the next series of electrode. And these growth cones will now grow in a very orderly and highly, so you can see these trophic molecules, these nanoparticles are being picked up by the growth cone. So the idea is that if we could allow these brain cells to grow in an orderly fashion, imagine a tube whereby brain cells are now growing along the lumen of the tube first. And once they grow, the other axons will adhere to them, and then they grow along those axons. So you put the traffic lights, you make the growth highly ordered, and you also prevent any clumping of these processes, and you promote nerve regeneration, which is faster and much quicker recovery, and there is no neuropathic pain. So we actually have a patent for this, a couple of patents, and we are working with Integra Life Sciences to be able to come up with tubes that could be implanted after nerve injury, whereby we will be able to guide these axons in a highly ordered manner. And you can see that as it grows along the lumen, each and every electrode has a capacitor. So you think about it has a capacitive charge. So when growth cone makes a contact with this series of electrodes, it changes the resistance of the electrode. And that electrode is then switched off. But in a series, it will activate the next series of electrodes. And when these electrodes are activated, they will create an applied electric field or uncaged trophic molecules. So you do systematically whereby the growth cone, it's a fully automated process. You don't have to do anything. These electrodes are smart. They will detect when the growth cone makes a contact. And the moment it makes a contact with that particular, the actual capacitor resistance is altered. It turns itself off, but then it turns the next one in the series on. Now imagine this thing turn into a tube, and this tube could really be placed between, as a nerve graft between the peripheral target or another nerve, and then you bridge the gap between them. So this is really work in progress, but I think that if this approach becomes really fruitful and in a clinical setting, it will also offer a tremendous opportunity for spinal cord injuries and also many other aspects of it. So this is really the first part I showed you, is really focused on nerve regeneration and how the semiconductor chips or these, this nerve conduit, a chip conduit or an electronics conduit can play an important role in tackling this problem of incomplete mismatch innervation and also the neuropathic pain after injuries. So this is work in progress. And the final part of my talk is really focused on brain-machine interfacing and how can we really use this approach to now understand how brain functions and what happens if a brain is damaged. We know the brain is a biological, of course, when you look at a stroke patient or somebody who had meningitis, the kids who died of any other injuries, this lump of tissue that sits in front of you, it's primarily biological. We know it has neurons, as I showed you, it has glia, it has all the other things, and different parts of the brain do different kinds of things. But this is not what really creates our thoughts, our feelings, our beliefs. That is mostly electrical, and it is not observable or measurable by just looking at the tissue itself in its own self. So how does brain function? It's really important, and of course tens of billions of brain cells. I started with 12 neurons, and I thought that I walked in water, only to realize I was the dumbest person on the planet. Recording concurrently from 12 cells. Now people are recording up to making contact up to several thousand, 4,000 neurons, and even larger neurons could be recorded from. And so the idea is to really, if we are to understand how brain functions, we must acquire the ability to record from large networks of neurons. And that has been one of the holy grail of neuroscience, the grand challenge to be able to record at a single cell level. The other grand challenge in neuroscience is, I think this is where the paradigm shift needs to come, is that the damaged brain tissue cannot be repaired. Stem cells are not going to work. They turn into tumors. And even if you produce those neurons, the guidance molecules, the ones that pave the road, are no longer there. And as a result, you cannot get these neurons to grow from one place to another place, because all those molecules that pave the way have really gone away. So can we then now use these brain chip interfacing conduits to be able to regain lost brain function if we were to create a hybrid whereby a brain cells can talk to a semiconductor chip, or a chip could talk to brain cells? And we know that the brain seeks purpose through interconnectedness. And brain cells, if they don't talk, the cells that fire together, they wire together. If they don't fire together, they will not wire together. If they, kind of fire apart, they fall apart. So we know that the electrical activity really plays a very important role. And if that electrical activity is no longer there, the brain cells really lose purpose in life, and then they commit suicide. And that happens with chronic depression and many other things. So we don't really have the means to repair brain wiring once it has gone haywire. We try to do something really very, very important. So they asked the question, if communication is really important, for example, if you have a kitty cat, early stages of development, you put a patch over one of its eye, and a few days later, you remove that patch, the kitty cat is blind in that eye for life. So the sensory input is really important for visual tactile pathway development. And in its absence, brain cells just do not connect. And this deficit then remains with us for life. Children who cannot hear, they cannot speak. So I think, this interconnectedness and communication is really important. So we need to understand how they communicate with each other. So we know the synaptic transmission, the conventional way we could record from brain cells is either using, sharp electrodes. We could use patch electrodes, and we could, understand what it is that forms the neuronal excitability basis of brain connectivity. When you fire action potentials in one cell, you can record one-for-one excitatory postsynaptic potential in its connected cell. You fire a burst, you get a compound response. And then when you fire a single action potential, you see that the actual amplitude of this EPSP is much larger now than it was before the burst. So this short-term plasticity, because it returns back to short-term plasticity, is also really important that we need to keep in mind. The brain cells are not hardwired. They undergo dramatic plasticity. This plasticity forms the basis for all learning, memory, and adaptability by our brain. So not only we have to be cognizant of the electrical parameters, the ion channels, the neurotransmitters, the receptors, but we also need to know that this is not a hardwired system. It actually undergoes synaptic plasticity, and it changes all the time. So this change is also really important, but it also is a treasure for us because we could alter, it's not a fixed action pattern. So what we do is in our lab is that we can actually grow these brain cells on a chip. We can record their activity patterns and for days and weeks, and to be able to really do a true brain chip and a brain computer interfacing, we needed to make sure that we develop a biocompatible substrate. We develop electrical components that are sensitive enough to be able to record from individual brain cells. And then to be able to not only have one-way talk, like you would see for epilepsy patients, where you're only recording the discharges and you cannot interfere using those same components. Or in Parkinsonian patients, you insert these deep brain stimulation electrodes where you stimulate those neurons in the substantia nigra and then get the dopamine cells activated, but there's not much you could really do. in terms of getting a feedback from those cells. So that would be the situation here. When you see that stimulator can stimulate a neuron, and deep brain stimulation, neuron is screaming and yelling, stop, stop, nobody listens to it. The other way I talked about is in epilepsy. When the neurons are firing, the transistor picks up the activity, but there's not much it could do. So what we wanted to do was to, instead of using a direct electrode to stimulate that you use in DBS, to use a capacitor that could stimulate neurons the way they are activated naturally. And when it fires, it will activate its partner, and underneath is a transistor that picks up a signal. So you complete a loop whereby an electronic device can talk to brain cells and they can talk back. So in instances where you lost the connection between these two brain cells, you can bypass this by using electronics to, for example, stroke had destroyed this part, and then these brain cells were disconnected from sensory to motor, or motor to other areas. Now you can bypass using this technology. This, I still think, is a neuronal dynamics, and digital computation is still a bit way off. So the way it really works is that you have, in our original prototype chip, was that each and every electrode had to be connected with this male-female component, and we could create a capacitive charge underneath the brain cells. It depolarizes as if another brain cell has activated it, or an impulse has arrived. So if you use the same capacitive current, the brain cell fires, the impulse travels along the axon, it activates the connected postsynaptic neuron. And when that postsynaptic neuron fires, you have a transistor, you pick up the activity of that cell. So you complete a loop whereby an electronic device can talk to brain, and then the brain can talk back to that electronic device. And of course, you know, these brain cells grow beautifully on the chip. They don't mind at all. There is no repulsion, there is no rejection. And that was a critical, really, chemical engineering component, because without this, it wouldn't have really worked. We also had to keep the brain cells alive. So we developed an incubator that has CO2, PCO2, temperature, humidity control. Everything is controlled, and it is cell phone and remote controlled. We can monitor cells, record, and maintain everything while sitting at home. And when you grow these large networks of brain cells, you can actually monitor the activities from days to weeks to months. And that was prerequisite to understanding how brain cells, you know, connect with each other, how they, once they're connected, what is their activity patterns, how do they develop networks, and can we record the network activity? But still, they're cultured neurons. We wanted to go to intact brain slice, and I really didn't think that I would live long enough. So you have capacitors here. Capacitors will stimulate. This is toxin. Let me play it back and slow it down. So what you're going to see is a hippocampus slice, whereby where you have CA3 and CA1 area. So this is the presynaptic area that needed to be stimulated. This is where the input comes in. And when these neurons are activated using capacitors, they will activate postsynaptic neurons, which are located in CA1 area. So you then put transistors in this area, and you have capacitor in the stimulation area where presynaptic neurons are, and you can actually record and monitor the activity of the entire circuit and alive. So when you have toxins where you only activate presynaptic cell, and there is no activity that projects to postsynaptic cells because you have blocked using TTX, the actual electrical impulse traveling to the postsynaptic site where it can activate postsynaptic cell, the activity will die down right here. Or you can use NMDA receptor blockers, or you could use really the receptors on the postsynaptic site. Without the toxin, what you're going to see here is when it is all in real time. So when you stimulate these neurons here, you will see that the entire slice lights up. And here we go. So with toxin, the activity really dies off. It doesn't go anywhere. But without toxin, you could see the presynaptic neurons are activated. They will activate postsynaptic neuron, and then you record from the entire slice real time using this hybrid of capacitors and transistors from an intact slice, brain slice. And so when we take the freshly isolated, even with our 3D electrodes, we design these electrodes so they actually penetrate into the slice, and they could record activity at the single cell level. And you could see the actual spread of activity. And this is the induced seizures using, either applying potassium or using a knockout mouse model which spontaneously seizes. So the idea is that you can actually record this activity in an intact slice, because that's really important, because these are naturally assembled circuits, unlike the cultured paradigm where they are unnatural. We don't know if two brain cells will ever be connected with each other and in the fashion that they connect in culture, because when you take parts of the brain, there are millions of cells, you dissociate them, they just randomly connect, and they may not be connected though that way in the intact brain. And here you have really an intact slice, and you can record the activity from large networks of neurons. But there still is a problem, and the problem is we are only recording from neurons that are juxtaposed or are in contact with the chip. Brain is three-dimensional. So what about those neurons that are sitting in different levels? So we grow these brain cells in silicon wafer, and this silicon wafer, we also label these neurons with fluorescent dyes. It could be either a voltage sensitive dye, or it could either be a calcium indicator dye like Fura2 and others. And when we fire a laser through the microscope, laser changes the properties of this wafer. And then when the properties change, it will activate that particular neuron. And once that neuron is activated as a presynaptic stimulation, it will activate the entire network that it is connected to. So what happens in that case is the following. You fire a single laser, you could see the entire network really lights up. And you're now recordings from up to 100,000 or even up to millions of neurons that are not just in immediate contact with the chip, but they're thinking in the entire slice. So the idea really is that, can we now take this technology into a clinical setting whereby we could now have the ability to monitor, directly stimulate brain cells, and then monitor the activities of large networks of brain cells, indirectly. So you can imagine that if in animal model, if we were to plant a chip in the sensory motor cortex, when the animal makes a contact, for instance, with the food pellets, the visual cortex will talk to the sensory motor cortex, and it would have to reach out normally to grab that particular pellet. Now imagine a chip that is implanted in the sensory motor cortex, which acts as a remote control. And because you will design these prosthetic limbs yourself, you could pair them just like we pair our remote control with our TV and other devices to be able to really have these brain control devices. So the important thing really is that as this technology mature, it had really tremendous potential, not only for understanding the brain circuits of the brain cell functioning, but also to be able to really use a true neuroprosthetic device that could be controlled by using this approach. So we, the last couple of my few slides, we wanted to really take this now research from bench in the bed area, and we targeted first the seizure detection. As you know that, especially I was the director of Children's Hospital, when kids don't respond to any medication, they are they need to go through a surgery to remove that part of the brain where these seizures are coming from. But when these surface electrodes or depth electrodes are mounted, the resolution is really very low. And the other thing is that these kids need to be attached to a 30-foot cable. They stay in the hospital, and the device is also really quite cumbersome. So we thought that we could use our chip, which is 300 times more sensitive than conventional surface electrodes. And we also wanted to make it completely wireless, so we could send the kid home. And then we also wanted to make sure that the chip that we plant in these electrodes are MR compatible. So once these electrodes are mounted in the scalp, we could take an MRI image. Now we know the juxtaposition of these electrodes exactly in relation to different parts of the brain that we are trying to really determine or detect the seizures that are coming from. So biomedical engineering and our neuroscience teams, we get together. These are conventional electrodes that are used to monitor. You can see that these wires are attached, and then the kid will be attached to a 30-foot cable. They stay in the hospital, and there's a lot of cost associated with it, and also lots of other related tension and stress for the poor kid all throughout the time. So we wanted to do was to develop a chip that could be implanted in the brain and then make it MR compatible and make it completely wireless. So these little sensors would then send any particular, every time the child really seizes, it sends a signal to a portable device that is either in the pocket or in the backpack. And it will not only provide the exact location of where the seizures are coming from, but also the incidence as to where, how many times or how often, excuse me, these seizures are occurring. Put it back together. And then what we have been able to actually do is to detect these seizures with exquisite accuracy. And then the detection is almost 50 to 20 times higher than the commercially available electrode. So we are being really approved to go through now the process of really testing it in the clinical setting is about to begin. So I think in the interest of time, I'll stop here. I'll be happy to take any questions.
Thank you so much, Dr. Syed. If anyone has any question, either you guys can type in the chat box or you guys could unmute yourself and ask the question. And I can go ahead with the first question. So during your first half of the presentation, you were talking about peripheral nerve injuries. And in medical school, they teach us that, either the peripheral nerve injury could be due to loss of myelination or due to, injury to the axonal branch or, along those lines. And then the most common cause for myelination injury is diabetes. And then the second most common cause for axonal injury is alcohol. And what you have done so far is trying to regenerate the axon by just guiding them to the capacitor and the transistor, like where they would be going to help with the neurogenic pain, right? But then what about, would it help with making the axons go and innervate the muscles so that they could restore the sensory and the motor abilities for the nerve injuries?
Yeah, so I think that really is what the ultimate goal is. So what happens is that when you have the injury, all these axons will try to grow at the same time. So they get really mishmashed. They often get cross-innervated. And this cross-innervation, slows down the entire process of regeneration. And regeneration also becomes incomplete and sometimes accompanied by neuropathic pain. So the idea really is that, whenever the nerve is injured, the environment alters quite significantly. We know at the injury site, when the calcium influx activate calcium-dependent proteases, which then chew up the axon, right? And if the muscle is not innervated for all that time period, it stops releasing the trophic molecules that will eventually guide this. So what you could actually do is to provide that supplementary, kind of material for it to, not muscles to stay active, not to lose hope. So you can also apply an electrode at the peripheral side, at the target site, or the muscle to keep it activated so it's healthy. It continues to release its recognition molecules or trophic molecules, so the incoming fibers can actually innervate or regain innervation. So not only, I think, the peripheral injury with our 3D models that we have developed, we are also using central neurons to study the myelination process. And we don't know if the Schwann cells themselves change or if it is the axon no longer interested in pairing with that particular Schwann cell. We don't know if the repulsion comes from the cell body or the axon or the Schwann cell itself. So we are collecting blood from these patients who are suffering from MS. We are, taking those different factors, separating different components and molecules that are, non-conducive to growth. So introducing them one by one and find out where lies the real problem when you do not get, these, Schwann cells just strip off, or oligodendrocytes in the brain just strip off and it leave the axon completely, naked. So the idea really there is that if we could really speed up the regenerative process, the muscles will not go through disuse dystrophy. The muscles will get the innervation. You can, enhance innervation many, many fold faster. And as a result, the actual regeneration can occur much faster and complete as well.
Thanks for explaining this. I think we have Taimur Bhai over here as well, who is also our executive member.
Assalamu alaikum, Dr. Syed. Thank you very much for your great talk. I'm so much, we're so privileged to have you. We're excited from your merit talk, and I was waiting to hear somewhat about knowledge mal. I hope all work is also in progress, but I have one question for you. As you know from the surface EEG, that we need to have a particular part of the brain, 0.6 centimeters, to be involved to pick up, to have the seizure picked up, or intractable discharges to be picked up on the surface leads. Regarding this Bluetooth new the product, the one you're working on, do you think it will be even helpful for the surgical patients, or we'll still have to go for, as you showed the pictures of the depth electrodes? So I was wondering how much is the catchment area for this?
Great question. I think you're stealing all my secrets, but we will tell you what the really idea is. The idea is that, can we now convert these electrodes, and we are actually fabricating these electrodes, whereby not only that we are using them as a detection device, these are 3D electrodes, so they can penetrate in surface depth, but they also anchor very, very good. And then the tight seal allows us to almost 15 to 20 times much greater sensitivity than either the depth electrode or the surface electrode. So we are pairing transistors and capacitors together. The moment the transistor picks up that there is an abnormal patterns of activity, which we program it, with the seizure like an electrical storm, it activates the capacitor, which is right adjacent to it. So now the capacitor will pass negative hyperpolarizing current. So to push this tsunami of seizures back and not allow the spread of seizure all across the brain. And we are also working, and we are actually making a deal with iPhone 18 and 19, whereby to have a cochlear implant that is sensitive enough to pick up seizure, and it dials your cell phone, and it tells you immediately, few seconds before you're about to have seizures. And Naweed, you're about to have seizures, sit down. I just dialed 911. I have turned the GPS locator on your cell phone. The ambulance is on its way, and you should relax. And I also told your wife, that you're richer than you think. So the idea really there is that, can we now take these technologies, they're becoming so sensitive, not really from a detection device, but also make them interventional. So you don't have to really do the therapy. The other thing is, when a surgeon resect part of the brain in a child or an adult where the seizures are coming from, in children particularly, 5 years post-surgery, these seizures return 40 to 50% of the cases. And because these kids underwent surgery, because they did not respond to drugs, what we are also doing is that we have set up a recording facility in our OR, and we often bribe our surgeons, with a nice desi lunch and, or dinner. And they spend some time to give us a nice slice of the tissue, which is intact from, the temporal lobe or wherever. And we take that immediately, we put it onto our chip, and we can now record spontaneously occurring seizures. But now we have almost 90 well, perfusion system. We can perfuse the drug to test which particular drug blocks seizures in that particular brain. And I was talking to Adil about precision health, or personalized health. Now we know that this particular drug X, Y, and Z, this concentration, this combination blocks seizure in this child's brain. Fast forward it 5 years, when the seizures return, the doctor can actually tell the family to use these drugs, and they will block seizures in that particular brain, right? So I think it's a multifaceted approach whereby not only that you are able to monitor, record, or do the intervention, but also to come up with drugs that will block seizures in that child in future, so you don't have to do another resection and another tissue. The issue is that it's mostly guesswork. When neurosurgeons, I've seen them operating, and they will go and say, my electrodes, these are 32, 35, 36, this range. I don't know, maybe the adjacent tissue will also be, they take out that part and then take out extra part. So that really ends up debilitating that particular patient for life. Of course, there is no choice for you, but they will take out often the normal tissue as well. So, but if your detection is really has exquisite accuracy, you could only target those specific areas where the seizures are emanating.
Thank you very much. Just as a follow-up to these things, to close it. So hats off to your work, because I was never thought of that it could turn out to be somewhat in a therapeutic way. So in the same line, I just wanted to ask you three things, and I think there are a few more questions. Dr. Taj Khan has a question, and someone in the chat box also asked a question. That we know that as a neurologist, we do all the time long-term EEGs, especially in the current world, EMU versus ambulatory EEG. So I was just proposing, I must be already in your pipeline, but something if the ambulatory EEG world and the business to it, and especially looking at the things back home, where I always thought that, how can we make long-term EEGs possible in Pakistan? So I was wondering, like, with this somewhat device like this, could be a very easy, either an add-on to the ambulatory EEG setups, or whether it is a replacement to it. And also if you can shed some light on there was a recent development going on with the endovascular interventionalists, that they were planning to throw some endovascular leads, EEG leads, for the long-term monitoring to kind of make it less invasive rather than opening up the skull. So if this technology, inshallah, as it looks, seems to be very promising, most likely there may not be much of a need of even doing the endovascular EEG placement. So these 2 points, and also if you can shed it, and whenever you get time, to if any progress happening on the knowledge mall aspect, is something very close to my heart.
So I think those are really great questions. And I think those are also very pertinent things that are on the radar. I think once you reach a level of sensitivity whereby you could monitor the activities of these brain cells, unfortunately, I didn't have time, but I did show it to the APNA group during my last talk, that you could monitor the activities of your sensory motor cortex neurons, without surgery, just by surface electrodes outside, and whereby we can drive a car, we can fly a drone. So I think the electrode and the technology is becoming sensitive enough that you could use them, for example, in ambulatory care, or even for very, very sensitive recordings, on the go. So I think the work is in progress. The field is heading in that direction that you pointed out. We are also working with Al Khan University, the neurology department as well, to trying to see if we could actually start implementing these technologies in Pakistan as well. So I think, give us a couple of years, we will hit that milestone. The issue really is that when you begin to record from large networks of neurons, often deciphering what is normal, what is abnormal, what is the aberrant pattern of activity, because it varies so much from, we have had rats implanted with electrodes for 8 months, and we never missed a single seizure. And even in the mouse knockout models, we never missed a single seizure. They seize all the time. So I think the sensitivity at that level is becoming quite very good. I think with the passage of time, you may not even have to implant electrodes in the surface or depth electrodes to be able to detect seizure-like activity. So I think, and I think same could be true for endovasculature or microvasculatures, or whereby you could perhaps use similar technologies to see other perturbations in the brain and the brain circuits. And the last question, things are going well. You know, as you know, that Islamabad area has a shortage of water, and that was a major concern for me personally, because water shortage is a really big deal. And when we started a couple of months ago, actually brought in the big bulldozers, and we were flattening out parts of the mountains to make way all the way up to top. Alhamdulillah, there was 8 or 9 fountains erupted, and we have 100 years of water supply.
Wonderful.
So, you know, I do sajda every time I see these kinds of things. So the Almighty is very, you know, kind to us and merciful. So some parts of the construction have already started. We had to really hire an international company to do the land survey because it's an earthquake zone. We needed to make sure that the animals were taken care of. We have cut into the mountains to actually create the animal housing, whereby we will keep all kind of exotic animals. You have the opportunity to even primates and all the other animals. And the construction phase is completed. We have been in contact with Imran Khan's government. He has assigned 2 teams to work with us to declare it as a research and innovation zone. It's a special zone whereby we had to negotiate that no money should be stopped because, you know, we will collect funding from abroad. From all good people, you know, and the idea will be to, you know, make sure that our state bank doesn't stop any of those payments. And you know how the inquiry goes, and you send a penny, and people are not interested in. So we got the clearance from the government. And the other thing we wanted was 10-year tax-free, 10 years duty-free for all the firms that are coming on board to set up their shop. So it's not just the knowledge mall, it's really the, you know, one-stop shop, but we are bringing in industry on board. So you don't even have to really cross the road to take your technology from prototyping to testing, to validation, to fabrication, and to production. So all in one site. And our Quran Center, Quran Research Center is almost complete. So it's going through the finishing touches. That is the first thing we wanted to make sure that, you know, we have an integrated, this concept of that science and Islam are not compatible. We wanted to kill it right from the get-go, that, you know, that these are both very important and the guiding principles, and they're both there. And then do research also on what we have been indicated in the Holy Quran. You know, there is no disease for which there is no cure. So can we actually create that mindset where we bring up this discipline? So some parts of the construction are already there. Government has given us over 1,000 olive trees we're protecting, preventing the you know, sanctity of the place. It's a huge land. We are also dealing with, and Imran Khan Saab has also announced a safari park in that region, which will be really very nice as well. So, you know, and then right adjacent on the other side of the road, the Beria town has agreed to build a university village whereby all the accommodations will be there for faculty, students, and housing. So we don't have to really spend any money. So the partnerships are coming together. But you know, in Pakistan, they always say, "Jab tak aap paiya nahi lagayenge, kaam nahi banega." And I am not really used to that paiya. And it took me a while to figure out what does this mean, paiya laga dein aap. So hopefully, I think the nation will take a lot of pride, but we will invite all of you to really take part in this. It is our national pride, is a national treasure. I had a grilling Dr. Atta Rahman just last week. And I said, you know, if nothing goes into the pipeline, nothing comes out at the other end of the pipeline. And you're talking about this, you know, this thing and that thing, where there is no receptive capacity. So alhamdulillah, we actually have now 70,000 students that we provide education for. There are 70 schools that we have set up through our trust. And I think that is the next step, that we will bring our own, you know, curiosity-driven, not degree-driven mindset. And we will create independent thinkers. I have now 5 students from Pakistan. They're all gold medalists. And you know, this lady was reading a novel, and halfway through she says, if there is a hero in this book, I hope he kills the author. And so, you know, they can't think on their feet, even though they are gold medalists. So I think his education system does not produce those independent thinkers. So the idea will be really to have these kids every Thursday, they will spend time in our facilities, and we will gauge their interest in different disciplines, and then find out what their core interests are, and then we actually put them into that pipeline. So stay tuned. Hopefully, I think once this COVID restrictions finish, I will get back and you know, get the project rolling. But we need everybody, all hands on deck. You know, अक्सर लोग कहते हैं कि आपको क्या मुसीबत है, आराम से बैठे हैं यहाँ पे, क्यों इस मुसीबत में पड़ते हैं? तो मैं उनसे यही कहता हूँ कि दश्त में प्यास बुझाते हुए मर जाते हैं, हम परिंदे कहीं जाते हुए मर जाते हैं, हम हैं सूखे हुए तालाब पे बैठे हुए हंस जो ताल्लुक को निभाते हुए मर जाते हैं. You know, at the end, it is our motherland. This is our country. हमने वहीं से baby step वहीं से लिए हैं, you know, and it's a payback time. So if we could really pay back and raise the tide so all boats come up, hopefully we will be able to do it through this particular initiative, inshallah.
Wonderful. Thank you very much. Alhamdulillah, inshallah, you'll do great. We all are your collaborators.
Jazakumullah khair.
Dr. Taj Khan has a question. And
do you have time?
Assalamu alaikum, everyone. I'm a pediatric intensivist. I'm not a neurologist. Sorry, I joined late. I found out when Temur posted on United Upna. But it seems like an excellent lecture. But I have some questions, clinical questions. Is there any research going on for a polio patient or peripheral neuropathy? There's just some quarters, you know, sensory, motor, or whatever, you can put it here, they can control and they are able to walk and talk. Or Guillain-Barré patients, how can we support them until they improve? So is there any research going on those fields?
So Dr. Khan, those are really great questions. And I think that is really the ultimate. In my last talk, I showed that how could you really bypass the affected area, whereby, for example, if you have a prosthetic limb or artificial limb, you know, we are natural cyborgs. We connect with devices naturally, automatically. You know, you're connected to your cell phone, it's connected with server, it's connected with satellite. And oftentimes when you have phantom pain or phantom limb, your brain still thinks that the organ is actually there, but it's not there. So can you now harness that power and have it control an artificial limb? And I showed the video last time that in front of a student, we divided and prevented them from seeing their own left hand. So they couldn't see their left hand. Instead, we put a rubber hand in front of them. And we stroked that hand with a brush, both his own hand, which he could not see, and the rubber hand, which he is focusing on. And then we actually bring a knife to hit on that rubber hand, and he moves his own hand away. I still have that video somewhere, but what really tells you is that your brain adapts. It can think about a particular organ that it is actually part of its own body. So the visual, the sensory, you know, perceptions that are coming from the right side of the brain, they will integrate. So I think for in the future, if you have the ability to link it up to a artificial prosthetic limb, you could control it by a device that you plant in sensory motor cortex, because you have a two-way link between a semiconductor chip and the brain. And when there's a two-way chip, you have almost replaced the damaged part of the brain, or you have integrated another component to your hardware, which now, because you designed it, they're talking to each other, it can become a remote control, which can control the prosthetic limb because you have paired them, you know, the natural cyborg idea. Mm-hmm. So I think in the future, the technology will be for children who lose their arms or lose their legs, you will be able to actually have them controlled through their own mind. And we control, you know, we can fly a drone, we can fly a car by just thinking about this. And these are the devices that could turn into, for example, for polio patients, for paraplegic, quadriplegic, and that you will be able to integrate these technologies. We are not quite there yet, but you have seen Elon Musk's technology, whereby they are integrating now a monkey is playing a computer game. And with the speed is just absolutely incredible. So I think this is where the actual field of neuroscience and neurology is heading.
Okay, great. And we would like to know about your organization, what you're doing. This is the first time I'm hearing your good work. So let us know how can we help?
I think this will be tremendous. And the unique thing about this particular project is that we are making a one-stop shop. So we have artificial intelligence, machine learning, robotics, and biomedical engineering, just the way the 8th through 12th century Muslims had in Baghdad and also in Spain, whereby we were producing polymaths. The people were working together to solve real world problems. So our education system has now compartmentalized everybody. There's a medical stream, this is engineering, they never see each other. We will create in that particular facility all these wings that are connected with the central dome, which is really the integrated learning area, whereby everybody will come and work together to solve problems that are complex. And we want to make our youth, our students, interdisciplinary rather than just one particular discipline specific. We are creating a dome, and that dome also has one of the largest Islamic era museum, and it's all glass, whereby children can actually work together and public can see from outside. Somebody says, I just developed a solar panel. Says, hey, can I use this for my artificial, a limb control, power for the retina development? This is a pretty cool because you don't need, too much heat, or you don't need this thing. So integrated approach will be the one, I think that's a part of that. So you have physics, you have life sciences from tissue engineering to genetics, and then astrophysics, quantum physics, and then chemistry. All these 5 wings are under one dome connected. So it's like a pentagon. And the idea really is that we will hire, all the directors will be overseas Pakistanis who have well-established credentials in the fields. No local people will be the directors. And we will provide them housing and travel. They come and they will say, 2 months ago, we agreed. You did not follow up, is a warning, next time you're fired. So no nepotism. All the teachers, we have now 3,000 already on board. They will come and teach a course for 2 years. And our local teachers will sit in the classroom and learn how to teach. Because, आपको पता ही, हम लोग 1930 के नोट्स हमें वही पढ़ाते थे. हमने मैडम उबैदा के नोट्स छुपा दिए तो उनको हार्ट अटैक हो गया बेचारी. So they didn't know what to teach. So the idea will be to bring these overseas, experts. They will teach. Local people can actually sit in the class and they'll learn. And so the idea will be to really take it a notch up, whereby nothing quite like this exists anywhere in the world. And alhamdulillah, we acquired the land. It's over 60-acre land. It's beautiful land. It's a landscape, is all hills and mountains. And each and every mountain will have a particular, building on it. But this is not my initiative. I can't do this. I'm not even a real doctor. You guys are a real doctor. I'm a phony doctor. And so the idea will be that if we could all come together and everybody will have their share. I'm actually talking to Bill Gates, one of the person in the contact that she seems to be turned out to be a Pakistani as well, running the chief operations of Bill Gates, that we will offer him for 500 million. We will name university after him, or anybody, any floor, any wing will be up for naming right. So if from Chicago, for example, people collected funds, that wing will be known as Chicago Wing, or the floor will be known as Chicago Wing, or the New York Wing, or the New York Auditorium. And we are building one of the largest library, digital library, which is on top of the dome, and it's all solar panel. The views are just spectacular. I mean, It just gets you in that mindset where you actually feel like you are in heaven. And I think that's what we would like to do, is to do that paradigm shift, inshallah, in Pakistan, and give something. Dekhe, maa to chahe diaper mein bhi ho, maa hoti hai. I know there are issues, there are problems in Pakistan, whatever, but still, it's our mother. And I think it's incumbent upon us. All jo hum apne mulk se chhod ke jab dene ka waqt aaya to hum chale gaye. But you know, we have very smart young generation. Pakistan's treasure is 54% of this under 25. So hoke mayus na angan se ukhado paude, dhoop barsi hai to barish bhi yahin par hogi.
Great. Other thing is that only 8 participants on this talk. So what happened? Why did we did not invite a majority or broadcast it to a larger audience?
Yeah, I mean, we did post it on the PINS group, and then maybe they are busy, so we have recorded the session and we would post it on the YouTube as well for them to kind of, you know, revisit the whole video if they want to.
It was a great talk. I think overseas Pakistani need to listen to this kind of a thought, and they should be involved, and I'm sure they will be excited to participate in those kind of projects.
Well, Dr. Taj, please do introduce us to the United Apna, this talk, you know, so we can have collaborators from your circles as well.
Yeah, sure. We will talk and arrange. I think this is an excellent thought and program.
Well, I think, you know, I have, you know, even if there are 8 or some people, but, you know, you have all been very kind and generous with your time. And that's really what it, in the end, it really matters. कि अगर 1-2 लोग भी आ जाएं, मैं अक्सर यही कहता हूँ कि, you know, the snowflakes are the most delicate things. You put it together, it becomes an avalanche. So I think if we could create an interest in our American and North American diaspora to really think about what it is that we could really do. You know, we have lived our lives, but, you know, Adil and these folks have a very bright future, and we have to leave it in very good hands. And I think that's really what the effort is. I never turn down a talk. I mean, I have 4 talks scheduled for tomorrow from, you know, women only groups and, you know, undergraduates and different groups. I am just paying my debt. I really have so much debt on my own people and nation that I am humble servant. So I don't really care how many people attend or not, but this is the debt that I am paying, and I will be held accountable at the day of judgment for this.
Great, thank you.
I think Dr. Chaudhary has a question.
Dr. Naweed Syed, this is Burhan here. Thank you for that amazing talk, really extremely inspirational. This is my 2nd time hearing you. Really, really love what you're doing, really love your vision, and really, inshallah, we all pray for your success. As a neuroimmunologist, I really hope that the roadblocks you mentioned when it comes to helping out patients with central demyelinating disease, I hope you can really conquer those and, you know, help these patients, because like I mentioned, I see a lot of MS, NMO, and the neuropathic pain can be pretty tough to treat. So I am rooting for you for sure. And as a neuro intensivist, I'm trained in that as well. I did want to ask you, I'm not sure if you'll be able to answer this or not, but the traditional process for the electrodes and their placement, we all know that it is a pretty invasive procedure as well. And since I staff ICUs, we have level 4 epilepsy center. So I see a lot of complications occur after those electrode placements. Have you, in your experience, seen that these surface electrodes that you are using and the surgeons that are placing those, have you seen a statistically significant decrease in these post-op complications, for lack of a better word, is what I want to ask? And then also, I believe your fellows run Neurora, the company, and the first time I found about you was through them. So they reached out to me, and they told me what they were doing, and I was fascinated. And that's when I read about you and saw what you were doing. So, you know, hopefully, I would love to meet you one day. I come to Calgary pretty much every year. So I would love to, you know, meet you one day as well, just as being, you know, a fan of your work. But good luck, and we are really rooting for you. Thank you for your time.
And thank you, Varun, for your kind words. And you just remember that you always have a home here. So, you know, whenever you visit Calgary, you're more than welcome to, you know, stay with us, or, you know, we will be delighted to host you and also show you around. So Neurora, folks, Pierre was my PhD student. He did his PhD with me. And now he's a postdoc in my lab, also supported by MyTech, which is a tech company. I'm actually chief scientist for a creative destruction lab, and which means that all the technology and that is going through the vetting process from, you know, validation to market across the country comes through us. And there are only two of us who are really the scientists. So Neurora went through that and it passed. We were also selected to represent Canada in the Falling Wall competition in Berlin. So a lot of really things that got us the best prizes, and in the epilepsy domain was that the non-invasive nature, the non-corrosive nature, the more biocompatibility issues, and also it doesn't really trigger the inflammatory response. So I think neuroinflammation is another major area. Whenever you plant these electrodes and either in the nerves, the neuroinflammation becomes a major problem because of the lack of the biocompatibility and other issues. So we have really made these flexible electrodes. They're just like your Dura that, you know, in some of the pictures you might have noticed, it is just like a Dura. It really covers up and it doesn't, it's non-invasive as your depth or surface electrodes will be. You know, so hopefully, I think, you know, we were scheduled to do full clinical trials before the COVID-19, then all non-essential surgeries were canceled, but we are going through the same phase. I was also the founding member of Neurosilicon, which was the first company that we set up, and we developed, you know, a number of techniques. I'm a very strong proponent of not securing. We have given a lot of licensing to Elon Musk as well, no questions asked. He's got much more money, and I don't really care if I benefit or not. As long as humanity benefits, we will pass it on, the licensing to them as a non-disclosure. So I think, you know, all of your questions are really important, and neuroinflammation for most of these invasive devices is a major challenge. You think about deep brain stimulation electrodes, they corrode, they stimulate neurons at a very high frequency. The neurons are screaming, stop, 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 but it doesn't really listen. And as a result, you get excitotoxicity, you get the damage in the brain area, and you also cause glutamate induced excitotoxicity. So that part of the brain actually degenerates, and these electrodes become less efficacious and effective in the longer run. So I think the more we go in that direction, the more sensitive the electrodes become, as you know, was said earlier, and the more non-invasive and biocompatible they become, we should be able to tackle the neuroinflammatory responses as well.
All right, thank you, appreciate it.
Dr. Syed, two last questions, and they are along the same lines for deep brain stimulation. So one of the participants asked, is there a way to stimulate the deeper parts of the brain with this technology to treat symptoms like depression? And then the second question is, with the same technology, is there a way to inhibit the unwanted signal, as in a spasticity or overflow phenomena, as in dystonia?
They're great questions. I think they are both a couple of $1 million questions. If somebody were to give me that money, I could probably provide them. But you know, the transcranial magnetic stimulation is capable of doing a lot of these things non-invasively. So when you do this transcranial magnetic stimulation to kids who have ADHD or hyperactivity, obsessive compulsive behavior, you know, when we put these electrodes and we zap them, it's amazing that what happens within 5 minutes, right? they sit down, they calm down. So you need to be able to really cut off the unnecessary hyperactive area, but at the expense of really enhancing other domains. But the problem really is we don't know what collateral damage these technologies do in the longer run, right? So brain changes, but it always at the expense of something else. So if you are stimulating or inhibiting a particular area, am I inducing structural and functional changes in parts of the brain that are not related to that function, but somehow connected? So in other words, you know, we use transcranial magnetic stimulation or mounting electrodes to prevent obsessive compulsive behavior, you know, ADHD or other kinds of aspects. We don't know in the long term, what does it really do? And we have learned, my lab also works on looking at the effects of anesthetics. And we know kids who are exposed to anesthetics early in their life, even twice or three, they have cognitive deficits when they grow up, because it's a critical time period when brain cells are connecting with each other and they're firing together, they're wiring together. If you prevent that firing, they don't wire properly. So the idea, but unfortunately, anesthetics are necessary evil, right? So you can't operate without them. Pain is even more, could be more disastrous for them. So how can we come up with approaches whereby we could either provide enrichment strategies or alternative ways to be able to really shift the brain activity non-invasively and not using those technologies? So transcranial magnetic stimulation is already being used for those kinds of parameters, and it seems to work, but we don't know what the long-term impact of this technology could be on the rewiring part, the plasticity part of the brain. And all of a sudden you stop someone from being obsessive compulsive, they become really, you know, something else, a serial killer or so. So the jury is still out there. I think once we have learned a lot more from those kinds of technologies, which is more like a shotgun approach, I think that developing these technologies that we are working on, you know, at a limited scale and a very well-controlled stimulation, perhaps could really pave the way for those kinds of non-invasive interventions.
Great. I think it's a good time to stop the session. I just want to say thank you once again for taking the time out and giving us a great lecture, even though we were a small crowd, but I really enjoyed it.
Well, one person is good enough for me. So thanks for having me. It's a real privilege, and you don't have to thank me. I'm just your humble servant.